Okay. Who wants to take? I was thinking it's kind of late in the semester, but it would have been fun if we had had like a daily wager going on whether or not my computer will survive this lecture. I did receive my memory in the mail. That's funny. I received my. Sorry. That was a joke in my head that I can share with you. So hopefully by next semester my computer will be working well again. Feel free to complain as much as you want about it. Actually, please don't complain about it. Because it's annoying. Okay. We're going to move on. Lecture 25. Quick business. Exam 3, the median was a 62 out of 80 for the multiple choice and a 20 out of 30 possible points for the short answer. So that's pretty good. We're happy. Okay, questions I answered last time. I'm answering again for those who are not here because they're panicking about the exam. Do I need to take the final exam? Not if you are happy with your grade. Do not come because you feel obligated to. Is the final exam cumulative? Yes, no, not really. It's mostly focused on this stuff, but be able to integrate the other stuff into it. Do we have a quiz this week? Yes, we're going to have another quiz this weekend because I don't want to. I'm kind of tired of writing quizzes. But I want to give you guys the option to drop as many low quiz grades as you can. Do we have recitation this week? Yes, the last one. So one last recitation quiz. And you guys have been asking me for your grades if you have to take a final, and I'm saying to you, well, right now this is where you are, but it all depends on how you do on the next few assessments. So, for example, if you're banking on dropping a quiz, you might want to take it. Right? Okay.
Question. I know you've probably been asked this a million times, but if you do a quiz this week, or does that mean you're going 3 on the quizzes, or just still 2?
For the weekend quizzes, if we do a quiz this week, it means we're dropping 2. Yeah. Because it's the best 10. Recitation quiz, we're always dropping 1. Yeah. Yeah. We will turn in our notebook. Right, thank you. Bring your notebooks on Tuesday, and I'll collect them. Next Tuesday. Okay. Let's take 5 minutes to write. Okay. I'll let you get your stuff out. Minute. Your papers. Okay, so you are studying. Are you studying? You're studying the effect of an experimental cancer drug on cell cycle arrest. So you want to see whether this drug causes cell cycle arrest. You decide to treat 2 plates of cells, one with drug and the other with a control buffer. You need your control. You conduct 2 experiments. One where you're doing BRDU labeling, and the other with PI staining and flow cytometry. And you get the following results. For the BRDU cell, labeled cells, your control cells, you get 30% are BRDU positive, and with your drug-treated cells, 30% are BRDU positive, and then for your PI staining, you get this data. So I want you to take a minute to, 5 minutes, to think about this and answer it as best as you can. Oh no, let me turn it up. I'm sorry. There you go. Okay, do you guys need more time or do you want to go over it? Someone said more time? Who wants more time? Okay. Okay, how are we doing? Good? Okay. Alex Mitchell. Alex? Okay, so tell me how you approach this. So there was nothing that came to mind. How about a keyword that came to your mind? You saw BRDU and you thought. Okay. Okay. Thank you. I appreciate honesty. Okay. Let me check you off for being brave. Somewhere. Okay. Who wants to tell me how they approach this? Who I never hear from? Yeah. Okay, BRDU is okay.
So, and I took that to the drug test, and I know the first case, I know it's for the cancer, but then the second case, I basically said that it's not good, that it's that or it's not going through cell cycle normally because the peak is still high.
Okay, so what I'm hearing is BRDU has something to do with whether the cell just went through S phase. So that's right. And so when we looked at the BRDU data, what did you see from that? That was. Well, so this is PI, but this is the BRDU. So it's the 30% that you want to pay attention to. So they're the same, right? So there's no effect on what phase of cell cycle. S phase. Okay, so that's what you can take away from that part. And so then you were talking about the different peaks in PI staining. So who wants to tell us more about how they approach the PI staining part of it? Do you know?
Well, I know that the first peak is the cells that are in G1,
Mm-hmm.
and like the swoopy part is S, and the next peak is G2. And in the drug-treated cells, there's more G2. So it can, what you can take from that is that the cells aren't going through cytokinesis, but they're duplicated. They can't split up, and then that's where the cell cycle is being stopped by the drug.
Okay, so, right, so the first peak to the left is G1, the swoopy part. That lower part between the two peaks is S phase, and then the other peak to the right is G2M. And the cells kind of cycle back around to that first peak as they go back to G1 after cytokinesis. So in the control cells, this is what you would normally expect to see, just in plain old cells. But in the drug-treated cells, the peak is larger in the G2M, meaning that the cells are not going through cytokinesis. So they're basically stuck somewhere in G2M. Did anyone have anything they want to add to that or questions that they had while looking at this? You can pretty much guarantee questions like this will be on the exam. So it's good for you to raise your hand right now. And it's recorded, yes.
Is there any comment on living a lot of cells in G1?
So the way to think about it is always comparing back to control, right? And so there are a lot of cells in G1, but there's always a lot of cells in G1. That's just what you would normally expect. Most of the time, cells are in G1, and for a shorter period of time, they're in S and G2 and M, which is why those peaks are usually lower. Yeah. Yes.
Can you go through BRDU again? When does that tells us that they're stuck where or?
So BRDU is a thymidine analog. What's thymidine? It's a base, right. So when are bases incorporated into DNA? What phase of the cell cycle? DNA replication, also known as S phase, exactly. So if cells are BRDU positive, meaning a lot of this thymidine analog is incorporated, then you could say those cells have recently gone through S phase, because it's the only way it's going to get in there. Yeah. So you kind of need the two bits of information because you want it, the BRDU is telling you something specifically about S phase, and then you're kind of getting a bit of a confirmation with the PI staining. And as long as the two bits of data jive, you're good. Okay, any other questions before we move on? Okay, so far, this list is going to be what's going to be on the exam. Motor proteins, muscle contraction, and row from the last set of skeleton stuff. And then the other day we did phases of cell cycle and checkpoints, and we started to talk about how scientists study cell cycle, and you just use some of the stuff we learned. We talked about conditional mutants, those were those temperature-sensitive mutants. Model organism used for studying cell cycle, BRDU, PI staining, and so on. So now let's talk about regulation of the cell cycle control system, because you know everything I talk about is regulation. So cell cycle regulation depends on the activity of CDKs, cyclin-dependent kinases. They're called cyclin-dependent kinases because they require cyclin to be active. So these are kinases that are going to control the activity of different proteins that are involved in controlling the major events of the cell cycle, right? So what do kinases do? Phosphorylate. That's getting louder, that's good. And so they're phosphorylating things that need to be activated. Great. So cyclins need to be present, and they need to bind to cyclin-dependent kinases so that they're active. Sorry guys, I'm just not feeling very well. No, fucking, we're having. Okay. Okay, so let's look at this. Let's look at this graph here. This is showing the rise and fall of cyclins. And so notice at different phases of the cell cycle, different cyclins are going up and then they're falling. Cyclins go up and then they fall. And so remember, cyclins regulate CDKs. So if a cyclin's not there, how is that going to affect the activity of a CDK? Right, CDK won't be active if there's no cyclin, right? So that's a key concept in cell cycle regulation. No cyclins, no CDK activity. And so automatically in your head, you must be thinking to yourself, oh, so these cyclins are fluctuating throughout a cell cycle, and that's controlling the activity of CDKs that are specific for phosphorylating specific targets during those phases of the cell cycle, right? So cyclins are rising and falling throughout cell cycle. They're activating CDKs and phosphorylating target proteins. So just an example. So if I say kinase, you think phosphorylate, you need to think to yourself, okay, so what does that mean? That means phosphorylation of these target proteins is going to control their activity, right? You're either going to have activation or inhibition of the protein depending on where it's phosphorylated or the protein itself. So don't worry right now about exactly how the phosphorylation affects whatever protein, but just understand that it's regulating its activity. So this is just an example of the huge number of targets of CDK1, which is a mitotic CDK. So if you look in here, you've got a bunch of categories of proteins that are phosphorylated by CDK1. Proteins that are involved in CDK1 regulation, proteins that are involved in DNA replication, proteins involved in mitosis, proteins involved in spindle assembly, proteins involved in actin polymerization, proteins involved in other proteins like DNA repair. Lots of proteins are targets of CDKs. Just like all kinases, there are lots of proteins that are phosphorylated by them. So there are many different levels of regulation of CDKs, and this is important because when something is important in a cell cycle, you want to make sure that CDKs aren't activated when they shouldn't be, right? So a few examples. So this figure shows it nicely. This figure right here. A few examples of different mechanisms for regulating CDKs. One is the availability of the cyclin, right? Just like I told you, cyclin needs to bind to CDK for it to be active. If there's no cyclin, CDK is not active, right? Of course, the availability of target proteins. That's the protein that's going to be phosphorylated. If that's not there, it also can't be activated. For example, proteins required for mitosis should only become available or to be turned on in G2. Then there's inhibitory phosphorylation of the CDK. So this would be phosphorylation that can happen in the active site of the CDK, so it'll block its activity. There's activating phosphorylation of the CDK. And then there's also CKIs. These are inhibitor proteins that are going to bind to the cyclin CDK complex and inhibit it. So this is how it looks. You've got your CDK, and it's got this loop here. Notice that changes conformation. So here's your inactive CDK, and when your cyclin binds, it changes the conformation, so it's partially active. And then once CAK, the CDK activating kinase, comes along, it can phosphorylate it in an activating way, change that conformation of that loop, which allows it to become fully active. So right now there's two criteria for activating CDKs, right? Cyclin binding and phosphorylation by CAK. So then you can have negative regulation of CDKs, right? you can have inhibitory phosphorylation. So there's Wee1 kinase, again, a kinase, so you have to think to yourself, it phosphorylates. What does it phosphorylate? Well, it's going to phosphorylate the CDK, but in an inhibitory way, in its active site. And so even though you have cyclin binding, that CDK cyclin complex is inactive because of that phosphorylation. But of course, phosphorylation is reversible. And so there's a phosphatase called CDC25 that can remove that phosphate group. And so then the other one I just told you about is a CKI, right? Cyclin kinase inhibitor. It's right in the name, so you shouldn't forget what it does. What it's going to do is it's going to bind to an active cyclin CDK complex and distort the active site of the CDK and keep it inactive. So those are the major mechanisms of regulation. These different proteins and kinases and phosphatases that are involved in determining whether or not the CDK is on. So the way you can think about cell cycle is that transcriptional regulation, so meaning upregulation of gene synthesis and protein synthesis, and cyclical proteolysis all regulate the cell cycle. Okay? What are some examples of types of proteins that you think might be degraded during cell cycle to turn on or off cell cycle or promote movement forward? So I just went through a list of proteins that are involved in regulating CDK activity, right? Yep. Cyclin. Right, so that's one protein that would be degraded. Yep. Another protein that you guys can think of? Cyclin's the most obvious one. That's a good one. CKI. Yep. And so what kinds of genes do you think might be upregulated at different phases of the cell cycle? So, cyclins. Right? If you're going to degrade it, you need to upregulate it and make it again so it can be available. Because remember, the cell cycle is a cycle. It goes around and around. So when you degrade something, you need to make more of it when it's needed again. Right? Okay. So this brings in ubiquitin ligases, right? These bind and transfer K48 linked polyubiquitin, so polyubiquitin chains, to proteins destined for degradation via the proteasome. We've heard about this before, right? So ubiquitin ligases help to regulate the cell cycle. And there's two we're going to talk about, APC and SCF. So here's APC. APC is inactive until it's activated by an activating subunit called CDC20. And once it's active, it can participate in polyubiquitination and degradation of different substrates. Some substrates of note are securin, which we'll talk about later, geminin, which we'll talk about later also, and S and M cyclins, which are some that we just mentioned. Right? So these ubiquitin ligases specifically target certain substrates for degradation. Questions? Okay, so the next ubiquitin ligase is SCF. And it's a different structure, a different kind of ubiquitin ligase. So this is a complex of proteins that contain a skip, colon, and an F-box protein. That's what SCF stands for. And F-box proteins are the specificity factor in ubiquitin ligase that recognize a specific substrate. And F-box proteins like to recognize phosphorylated substrates. So the example being shown here are CKIs, these cyclin kinase inhibitors. When they're phosphorylated by some other kinase, then they can be recognized by the F-box protein, polyubiquitinated, and then degraded by the proteasome. So that's one way of regulating. So here you've got multiple levels of regulation. You've got whether or not a protein is going to be degraded, and then you've also got whether or not your ubiquitin ligase is going to be on or not, right? Because with APC, that requires CDC20, so you need that activating subunit in order to degrade proteins. And then with SCF, you need to phosphorylate your target protein. Okay? Yes? SCF? So SCF proteins, they recognize phosphorylated substrates, transfer polyubiquitin chains to them, and then they can be recognized by the proteasome and degraded. And so the regulation of whether or not they're going to be degraded very much relies on that phosphorylation, because without being recognized by the ubiquitin ligase, you can't be ubiquitinated, you can't be degraded. Okay? Okay, so there are many different substrates of SCF and APC, and their the substrate degradation depends on ubiquitin ligase activation. If the ubiquitin ligase is on, so for APC, for example, if it's on and those substrates are around, those proteins will be degraded. And then if SCF also has to be active, and SCF has to be, then you also have to have phosphorylated substrates to be recognized. So if you look up here at this diagram of the cell cycle, you notice that different substrates, different ubiquitin ligases are active at different parts of the cell cycle. Okay, and so that's going to kind of have something to do with the types of proteins that are being degraded at the different phases of the cell cycle to allow the cell cycle to be regulated. And we'll go into greater detail about that. So, why so many levels of regulation? Why not just control cyclin synthesis and degradation? Right, you want many levels of regulation to avoid problems, because what happens when there are problems? Cancer. Right, exactly. And also cyclin synthesis and degradation, that's one thing, but let's say that gets dysregulated, then having that as phosphorylations and those other requirements in TKIs are just kind of another level of regulation. Right. So this is just a summary of the major cell cycle regulatory proteins that are in the textbook. You do not need to memorize these, but you do need to know them. At least the major categories. Right, so it's protein kinases, CAK, WEE1, CDC25, CKIs, ubiquitin ligases, and how they're activated and regulated. Okay, so many levels of cell cycle regulation, right? There's external effects, so the cells are sensing the external environment, right? If you've got an unfavorable environment, then a checkpoint will be triggered. If there's DNA damage, a checkpoint will be triggered. Same with, there's all these different checkpoints that will be triggered if the environment or the cell is not in the right state for cell cycle to progress. Then there's also control at the level of CDKs, right? If the cyclins are available, or if cyclins are available, or if the kinases are available, or phosphatases are available, right? And then there's levels at the level of transcriptional regulation, right? Because you need to make more of the proteins that you just degraded. You need to make more of these substrates that need to be phosphorylated in order for cell cycle to move forward. Okay, so you can think of this, these different levels of regulation of the cell cycle. Oh, and then of course activation of ubiquitin ligases, right? That's another level of regulation. Okay, so let's talk about S phase. So you can think of them S phase as being regulated by CDKs, so the S CDK, and by the APC ubiquitin ligase. And so we're regulating chromosome duplication. We need to make sure it happens when it should happen, not when it shouldn't happen. Right? What happens if you just keep on replicating your DNA before you go through mitosis? That would be bad, right? It's important that this is regulated and that chromosome duplication happens once per cell cycle, right? I see people nodding, that's good. Right, so this all has to do, regulation of initiation of DNA replication all has to do with ORC assembly and helicase loading. ORC, that's origin recognition complex. So that's this complex of proteins that's going to recognize the origin. That's where DNA replication begins. And then you load the helicase there. Because what does helicase do? Does anyone remember? It opens, unzips the DNA, exactly. So if you can't load your helicase, can you replicate your DNA? No. Exactly. So you've got your origin of replication bound by this origin recognition complex. And then the helicase and helicase loading proteins will be bound. That's called the pre-RC, the pre-replicative complex. And then SCDK will become activated, and it will do things like phosphorylate CDC6, which is part of that pre-RC, and you'll get degradation of that phosphorylated CDC6 by an SCF. Basically, what you're doing here is you're loading on your helicase, then you're disassembling that complex and recruiting your polymerase and allowing activation of DNA replication. This is all regulated by SCDK because you can get this assembly of this whole pre-RC and helicase is loaded, and it just sits there. It sits there and it waits. It's not activated until SCDK comes along and helps to disassemble some of these proteins, recruit polymerase, phosphorylate components of the replication complex, and then you get activation of DNA replication. So how does this assure that replication only happens once per cell cycle? Well, by disassembling this whole complex, right, SCDK is going to become activated, and then this whole complex is going to be disassembled. And without that pre-RC formation, without that origin recognition complex binding there and helicase being loaded, you can't start DNA replication again. So that allows the whole entire cycle to go through. And so you're going to say to me, okay, so what? Hopefully you guys can see the value in only replicating your DNA once per cycle, right? So this is important. So the other thing to keep track of is APC. That's that ubiquitin ligase. It is involved in degrading a whole bunch of proteins, but one protein I mentioned is called geminin. And geminin is an inhibitor of CDT1. So when geminin is around, this complex cannot set up because CDT1 is inhibited. It's being bound by geminin. And so that's also going to prevent DNA replication from kind of getting set up with an ORC formation. So once APC is turned on, though, geminin is going to be degraded, right? So, oops. And when geminin is degraded, that allows that CDT1 to come and bind and to bring in the helicase loaders and to load helicase, and then you can set up to start DNA replication. Right, so APC degrades geminin, the inhibitor of CDT1, when it becomes activated at the end of M phase. So at the end of M phase, APC turns on, degrades geminin, and now CDT1 is available for helping to set up that ORC and helping to get those origins ready for helicase loading and DNA replication. Okay? So this ORC, it needs to assemble. Otherwise, you can't load the helicase at the origin. And for that to happen, CDT1 needs to be available. And so during this, basically after M phase G1, APC is active and it degrades geminin so that CDT1 can assemble, bring in the helicase, and get that pre-replicative complex set up. So, like I was saying before, APC and SCF ubiquitin ligases, they're active at different phases of the cell cycle, and they have different substrates. They're degrading different things of the cell cycle. So what I was just talking about was APC and how it's turned on at the end of M phase, right? So all of these different proteins can be degraded, right? You see geminin in there. You also see some cyclins in there. So you'll start, as we go through this, you'll start to come back and look at this figure, and you'll see that different substrates that we're hearing about are degraded at different points of the cell cycle. The other thing I want you to notice is APC and SCF help to regulate each other. SCF is going to degrade an inhibitor of APC and allow APC to turn on, right? And that's happening, pretty much starts to happen in M phase. And then APC is also involved with activation of, or turning off, I should say, SCF. So there's this interplay between the different ubiquitin ligases, and you can kind of see each of them takes half of the cell cycle, pretty much. Any questions about this? Right, so now you've got your pre-RC set up and your helicase loaded, and then SCDK will become activated by binding bicyclin, and it can phosphorylate things. And one thing it phosphorylates is CDC6, which could then be recognized by SCF and degraded. And then you also have, now you also have APC being turned off. And that allows geminin to build up again. And so now geminin is going to bind and inhibit CDT1. Okay. SCDK also triggers the DNA unwinding and initiation of DNA replication. So that's pretty much what we're talking about. So you'll get DNA replication, and then you have your, then you can get DNA, then you can get set up of another pre-RC and get set up for another cycle of DNA replication once you complete it. But still, it won't be triggered. You won't have actual DNA replication until SCDK is activated in S phase. Okay? Okay, so this I just kind of go through all the steps again. So APC is active from M to G1, right, because it's turned on by mCDK, and this allows for the pre-RC to form. And then APC is turned off in S phase by the G1 SCDK, and this allows for geminin to accumulate, and that's going to prevent pre-RC assembly. And then pre-RC, the pre-RC will be disassembled, and DNA replication will turn on. Questions about this? Yes? So the question was, how is DNA only being replicated once? And that all has to do with whether or not your pre-RC can form. That's all of these proteins right over here that are loading helicase. And so if SCDK is active and phosphorylates components of this complex that can then be degraded, it's going to prevent that complex from being set up again and prevent helicase from being loaded again until that ubiquitin ligase is turned off, and then it can start to set up again in the next round of the cell cycle. Okay? Other questions? Okay, so here are a few cell cycle practice problems. So I want you guys just to take a few minutes here to go through this with your friends. How do cyclins regulate the cell cycle? You suspect that a defect in protein X has a negative effect on cell cycle progression. How would you use the tools explained in class to evaluate whether there's a defect in cell cycle and where that defect might be? What would be the effect of a constitutively active APC? So if APC, the ubiquitin ligase, is always on, how would that affect cell cycle? Or how about an APC that cannot bind to a CDC20, that's the activator of APC? And then consider an individual if they have a mutated FBOX protein. So their FBOX protein, which is part of the SCF, doesn't work. Would they be viable? Why not? So take a few minutes. See if you can tackle at least the first one, and I'll circulate and try to help people.
Yeah, it's just
Yeah, my grandparents, my grandparents, so they're 109,
landmark.
So excited. My boyfriend, she's younger than I am. So she was like, no, she was kind of yeah. But if you look at it though, the actual time itself is back. You know, there's so many things around it. That's like the same.
Okay. How do cyclones regulate the cell cycle? No cycle, no cell cycle. Okay, give me more. That would be 1 point. How do cyclones regulate the cell cycle? What do they bind to? CDKs. So they activate CDKs at the right time of the cell cycle. So each CDK is going to have its own cyclin. And so when the cyclin's available, it can bind to and activate the CDK. Right. If your cyclin CDK is inhibited by, say, binding by a CKI or an inhibitory phosphorylation by V1, it will also be on. Okay, so you suspect a defect in protein X has a negative effect on cell cycle progression. Okay, defect in protein X, negative effect, cell cycle progression. Okay, how would you use the tools explained in class to evaluate whether there's a defect in cell cycle and where this defect might be? Yes. You can use PI to see if it arrests itself in S phase, and you could use PI to see pretty much anywhere in cell cycle by looking compared to control, which has a normal protein. You would see if you have a larger peak showing up somewhere, like for example G2M. Okay, good. You could also use conditional mutants if you happen to have one. And then you would also be looking to see if there's a defective cell cycle stop somewhere. Other thoughts on that one? What would be the effect of a constitutively active APC or let's start with that one. APC that's always on. What's going to happen? Uncontrolled replication of DNA. Okay. Yep. Yeah. CDC20 turns on APC. CDC20 is an activating subunit of APC. So the constitutively active APC would, I don't even know how that would happen, but this is my question, so I will go with that. So you'd have to think to yourself, okay, what does APC degrade? Please tell me that's not me. Is it you? I'm always worried my computer's it's actually becoming an issue for me. Like I hear a noise and I'm like traumatized by my computer. So what does APC degrade? Well, one thing we just talked about is geminin, right? And so if APC is always on, then it's always degrading geminin, then that means CDC1 can't be inhibited, and you would always be setting up a pre-RC, and you would always kind of have the stage set for DNA replication. Now, that's a good answer, but it's not perfect because there's more than one level of regulation always. But that's logical thinking. What about an APC that can't bind to CDC20? An APC that cannot bind to CDC20, will it be on or off? Off. So this is an APC that's always off. That means you would always have what protein present that we just talked about? Geminin. Geminin's always there. It's always binding to CDC1. And so then you can never get setup of your pre-RC and initiate DNA replication. Yes? What would be the effect of always having APC active? So you have to ask yourself, what does APC degrade? Because it's using ligase, yes? Right, APC is always active, geminin's never present, then you're always going to have this setup for another round of DNA replications. Yep. Okay, what about an individual with a mutated F-box protein? Would they be viable? Could you even ever study such an individual? No, because what's an F-box protein?
It's part of the SCF.
Right, it's part of the SCF. What part does it, what does it do in the SCF? I know we just learned this. Recognizes the substrate, right. So if the mutated F-box protein didn't work, and so it either couldn't bind to SCF or couldn't recognize the substrate, would that substrate be more or less stable? more, right? It would never be degraded. And so what's one substrate we talked about with SCF? CKI. Cyclin kinase inhibitors inhibit cyclin kinase complexes from being active. So if you have mutated F-box protein, you can't degrade CKIs, and so you never can activate your cyclin kinase complex. Is that good for viability? No, you need cell division to be alive. Right? Okay. Good. Okay, so let's stop here today. And I will see some of you guys shortly.